Okay, so let's go ahead and pick up where we left off. Yes, I do have the exams. I'll give those guys back to you during lab once we settle in there. But let's go ahead and keep working on chapter 12. So in chapter 12, I wanted to talk a little bit about what Mendel saw and the work that he did. And I want to do this before we get too deep into the chapter, because what I want to do is I want to do a couple of things. First of all, so I want to correlate this with the scientific method. And then I want us to kind of basically take a look at Mendel's data and then kind of reason our way through what he saw. And then wrap up with basically, I'm going to show you how he did it, and that it's not that difficult, actually. You just kind of have to sort of train yourself to sort of think the way that he was thinking. So one of the things that I always like to mention about Mendel, I mean, obviously you already know my bias, right? This chapter is my bias, and I don't apologize for that. But one of the reasons why I like this particular section is because we talked in the first chapter about scientific method, right? So we went through the 6 steps of the scientific method. And really, when you take a look at Mendel, he was really the first person to kind of illustrate for us how to use the scientific method. Now, it doesn't necessarily mean that all the rest of us weren't using the scientific method, and it doesn't mean that he came up with it, right? Elements of the scientific method were already kind of in place. The chemists had a lot to do with that, and all that, right? But when you come to the 6-step method of the scientific method, Mendel's work with the monohybrid crosses is probably one of the clearest, cleanest, simplest, most straightforward representations of how to use the scientific method as sort of like a role model, basically, for us for the 20th century. In a lot of ways, when I am feeling relatively hyperbolic, I oftentimes will say that Mendel taught us how to do science in the 20th century by giving us such a clear example of using the scientific method and use very strong, rigorous, logic-driven thinking patterns to deduce data and to handle data. So in a lot of ways, he modeled for us what good research should look like. And it's very clean and simple and straightforward. So it's a really, really good one. So what are the 6 steps of the scientific method? Well, let's go ahead and list them out first. So we always start off with observation. And then we follow it up with question. And then we have our best guess, right? That's our hypothesis. So our best educated guess answer our question. And then we should be able to generate testable predictions, which we'll be able to with experimentation. Should have never stopped. Yeah, just saying. Make sure my participants are here. Elena's still good. Okay. There we go. So let's go ahead and take a look then at what Mendel did, right? So remember, Mendel basically kind of leaped off of where T.A. Knight left off, right? So with pea plants. And so Mendel did sort of the same thing. So he basically, I'm just going to take a look at back to work. So basically what Mendel did was he took two true breeders, just like T.A. Knight did. Darwin was a red flower. So T.A. Knight was the one in 1823 who started working with pea plants, but he didn't get as far as Mendel. So, and he crossed a purple true breeder with a white true breeder, right? And remember, we've already defined true breeder, right? A true breeder, basically, that you breed true for whatever that trait is. So in this case, a purple true breeder is only going to be able to pass on purple, right? Now, for a particular person like Mendel, what's the significance of this? Well, the significance of this is the fact that it sort of streamlines and eliminates variables, right? So whatever's going on in there, so as a particular person, your idea is that there's some sort of a particle instructions, if you will, for purpleness. And so what you know in this true breeder is that whatever is, there's only those purple instructions. That's it. So there's no extraneous unknown hidden variables. So it streamlines things considerably. So then he continued on and he crossed these two together to give him his F1 generation, which was all purple. So let's stop for a second. The first observation he makes is that they're all purple, right? So that's the first step of your scientific method, that F1 is all purple. So if F1's all purple, then what's your question? There's one glaring question. Okay, why is a good one? It's a little diffuse. It's a little broad, yes. Right. What's the glaring question if you're a particulate versus a blender? Exactly, right? The white. The issue is the white. Where's the white? So that's your question, right? So now you have two hypotheses to explain this, right? The blenders and the particulates. The blenders say what? The white is gone, right? Because they take blue and yellow paint, they mix it together and turns green. Your yellow's gone, right? Get over it, shed your tears, move on. You're never going to see it again, right? So you have the memory of it. That's all you've got. Pretty harsh, but right. But that's what the blenders are saying. Now, what do the particulate people say? No, it's got to be there somewhere, right? hiding in there. Right? So it's there, we just don't see it. Your hypotheses. And what about your predictions? So here's a good issue. If it's gone, then it's unrecoverable. That makes sense, right? Just like if yellow's gone, you're never going to see it again. Right? Notice the if-then statement. That's a prediction. However, number 2 also has a prediction, doesn't it? If it's hiding, then it's going to be recoverable. So these are your testable predictions. So then what did Mendel do with his experiment? He basically crossed the purple F1s with the purple F1s, right? He probably solved them. And then he generated his F2 generation. And what did he see?
White with purple dots.
Nope. That does exist. It's called variegation. Yeah, it's actually a different genetic mechanism that we didn't discover until the 1950s. Barbara McClintock discovered that one in plants. No, usually variegation in plants is caused by mobile DNA, at least one particular type of variegation, mobile DNA, where you have a pigment gene that just basically gets up, moves somewhere else in the genome, and then it just creates these like little spots wherever it lands. So you saw a 3-to-1 ratio. Now, the other thing I want to do is I want to insert another one. I'd love to insert my slides here. Because what I want to do is I want to make a list. It's kind of a glossary, a little bit. But this is going to be a list of Mendel's ideas.
Where was Mendel from?
He's Austrian. So this is a glossary of terms. And what I want to do now is I want to basically take a look at just this, just focus on this. And I want to reason through some of these ideas, because this also basically gives us a list of some of the major things that he was able to figure out. First of all, before he even set up a cross, he's a particulate guy, right? So he already believed that these traits are governed by specific particles of inheritance, right? And like sets of instructions that governs a gene or that governs a particular trait. We call this particle of inheritance a gene. And this controls a trait or traits. So for instance, our trait here is flower color. However, Mendel's a sharp guy. He's observant. You don't just have one flower color, do you? You got 2 different flower colors, right? So you got purple and you got white. Is purple a flower color? Sure. Is white a flower color? Is that controlled by one particle controlling flower color? Well, in his case, that was his hypothesis, that it was one gene, but you had different versions of it. Kind of like you have one baseball game, but you have 2 versions of it, American League and National League. Of course, that's starting to change now since we just picked up the DH, but in the National League. But you kind of had 2 different sets of rules, but you were basically playing the same game, right? Same game, 2 versions of it, right? And that's basically what he saw. What he saw was a purple version of the flower color gene and a white version. We call these alleles. So the example here would be purple versus white. But he also noticed something. First of all, he knew that white was inside the F1s. Why? Because it came back in the F2s, right? How could it just come from nowhere? Couldn't come from nowhere. So if it came back in the F2s, it means it had to be in there somewhere, right? So if you basically say that purple F1s have both purple, why? Because, well, you can see it, right? But it also has white because you can pull it out of there. Then what he pretty much said was that these two different alleles aren't the same strength. That there's two different strengths of alleles. There seems to be a dominant one, which expresses in one copy, and there also seems to be a recessive allele, which expresses in two copies and is masked by the dominant. So what does he know now? So what he knows is in my purple true breeder is only the particle for purple. And my white true breeder is only the particle for white. So what they must have done is they passed on a copy of the purple instructions from the purple parent and a copy of the white instructions from the white parent. So what he was saying is there should be white and purple in the F1s, but you only see one. So basically what he has is this idea of alleles. Now there's another thing that he also takes a look at as well. The purple parent and the purple F1, let's take a look at these two. If I were to take the purple parental and the purple F1 and mix them up, would you be able to tell me which one is which? No. Why? Because they're both purple. They're both same shade of purple. There's not a lot of variation there. They look pretty much the same. Okay, yes. You can differentiate them. We'll talk about that after this. Right, so now here's the next question. So they look the same, yes? Indistinguishable. However, do they behave the same genetically? No, right? Because the parental is a true breeder. That means it's only genetically passes on purple. But the F1 is a non-true breeder. It has the capacity to pass on purple and white. So what you have here then is essentially another set of terms. You have what we refer to as the phenotype. It is what you look like. Based on the alleles. And you have genotype, which is your allele makeup. At a gene. So now he noticed that you look one way, but you can behave another way genetically. Okay. Now here's the other thing, right? Mendel knows that the purple parental only has instructions for purple. Now I'm going to go ahead and sort of add a little bit of notation. He didn't do this, but intellectually, this is actually what he did in his paper. So if you basically take a look at this and you basically say that you get your DNA from really two people, mom and dad, that we've known for thousands of years, right? So if you get your DNA from mom and dad, that basically means that if all you've got is purple, then you've got two copies of those purple instructions. And not only that, but according to your observations, you've got two copies of the dominant version of this. Now, generally speaking, in genetics, dominance is usually denoted by an uppercase letter, in this case, P for purple. And so in this case, you have big P big P, because that's all you have to give. You have 2 copies of the dominant look. Same thing for mom, who has 2 copies of the recessive. So then the idea is the F1s get 1 copy of the purple from purple dad and 1 copy of the white from white mom. Therefore, you have both. Now let's take a look at this relative to the genotypes. What you see here now is basically 3 different genotypes or 3 different allele scenarios in any given place. The first one is going to be homozygous dominant. This is basically where a gene has 2 copies. Of the dominant allele, like that. You also have homozygous recessive. We have 2 copies of the recessive allele, like that. So that defines our parental generation, doesn't it? And then you have heterozygous. We have 1 recessive allele and 1 dominant allele. So this is basically your genotype. But the phenotype that's associated with this then is a little bit different, right? If the dominant allele, then your phenotype is going to look dominant. If you've got 2 copies of your recessive allele, then your phenotype is going to look recessive, right? If you have 1 of each, then your phenotype is going to look dominant. Okay, that's what Mendel saw. His F1s were purple, right? And they were heterozygous. So here you can basically see genotypes and their phenotypic outcomes. And Mendel basically got all that from his monohybrid cross. It's a really nice, because it's super simple experiment, right? I mean, we could have done this. All Mendel did was overlay on it scientific method and just looked at his data and just thought it through and just made observations and then kind of teased out the information. I like to make this list because it illustrates to you just how nice and how easy this can be sometimes when you just sort of look at your data and you just try to reason out and rationalize out what you're getting. Like when the heterozygote, you mean? Well, it's not necessarily white, whatever the dominant trait is, right? So in this case, the dominant trait obscures the recessive. It just happens that white is recessive. It's not always the case. In some flowers, actually, white is the dominant. And then the actual color is the recessive. And sometimes you can see this like in a plant population, because generally, look, and you can if we get kind of good at this sometimes, not always, but if you've got a good, healthy, homogeneous population that's well mixed, you can actually see the dominance recessiveness, because you can see roughly 75% of the population looks like this, about 25% looks like this. Visually, you're looking at roughly a 3-to-1 ratio. So you're seeing the dominant look versus the recessive. Then he obviously generated his 3-to-1 from that. So this is going to be your phenotypic ratio. And the reason why the phenotypic ratio is important is because these ratios are diagnostic. In this case, for the monohybrid cross. So if you're crossing something and you do Mendel's experiment, you see 3-to-1, it's a monohybrid situation, which means it's following Mendel's rules that he saw. We'll talk about some of those rules that Mendel found. There's also another one, however, the genotypic ratio, which is 1 to 2 to 1. Now, Mendel didn't stop at F2. He actually went to F3. That is to say, he went to the great grandkids, right? Because what he noticed was he crossed his parentals to get his F1 generation, and he crossed his F1 generation to get his 3 to 1. And he did this for a lot of different traits. We saw that. But now the question was, well, wait a minute, wait. I already see that not all purples are the same, right? That the parentals are true breeders and the F1s are non-true breeders. So now when I'm saying I've got 3 purples to 1 white, are all those the same purples? So Mendel wanted to tease that out a little bit, right? He said, okay, phenotypically you look the same. But now genotypically, that is genetically, do you act the same? Okay. And so what he did was he selfed them, and he saw that in one class you had a true breeder for purple. In 2 of the purples, you had non-true breeders. You generated your 3 to 1. So these are non-true breeders. And then of course, the white was a true breeder for white. So what he saw was 1 true breeder for purple, 2 non-true breeders, and 1 true breeder for white. 1 to 2 to 1 genotypic ratio. So what did he come up with? First of all, these are binary traits. So sorry, blenders, you're out the window. He also saw dominance and recessiveness, right? So very clean cut dominance and recessiveness. And ultimately, he looked at segregation. Generated, of course, is 3 to 1. That was a big one, right? So the observation of 3 to 1 was a big one. That was a huge one. It's kind of our elements of Mendel. And first of all, this kind of leads up to one of his big ideas. He had 2 really big laws in genetics, segregation and independent assortment. We'll talk about those here in just a second. But one of them was that you had discrete factors, right? So that is non-blending. Everything that he did was consistent with these particles, with these discrete factors. He also noticed that you get one of these, one from each parent. Right? You get one from mom and one from dad. And he also pinned down the allele idea, the alternative versions of the same gene, and of course, genotype, homozygous and heterozygous. Okay, so he pinned all that stuff down. So pretty much everything in our glossary list, he essentially organized. Now, here's the important thing about the list. Mendel didn't have the terms. So it's important to understand that Mendel didn't have the term homozygous or heterozygous or gene or allele. He didn't have any of that. That came later on. A later generation of geneticists invented those labels and terms and put them on there. What you see in Mendel's paper, which is fascinating to read actually, is what you see are the ideas. So what Mendel does is he discusses things. So like he'll be discussing something, and what he's describing, you realize is like he's describing like a homozygous dominant. That's what he's describing, right? So he describes these things, but he doesn't actually give them labels. Later on, we gave them labels. Of course, we already know that he gave us the alleles. We have dominant and recessive alleles, and of course, he gave us the genotype and phenotype, our physical appearance. Now, the big thing that Mendel came up with, his big principle or his law of genetics, if you will, is the first one of the 2 is the law of segregation. So what is that? So basically the law of segregation is that you have 2 alleles that are basically segregating during gamete formation, and you're getting one from each parent. That's his law of segregation. Now, hold on 1 second. There's a couple of things that we need to clear up and make absolutely crystal clear. First of all, Mendel didn't know what an allele was. The term didn't exist. He didn't know what a gene was. The term didn't exist. He didn't know what gamete formation was. Nobody understood what gamete formation was. We didn't know anything about mitosis at this time. We didn't know anything about meiosis. We didn't know anything about chromosomes. So we didn't have any of that knowledge. So Mendel didn't know anything about meiosis at all, or chromosomes, or cell cycle, or any of that. So then What did he know? What Mendel knew was information. So when he was talking about a particle of inheritance, what he was thinking of, and this is the reason why I like to use this term, he was thinking of a packet of instructions. Think about it. A packet of instructions is basically a little thing of information, right? That's the way Mendel thought of it. And for a long time, actually, for at least a couple of decades into the 20th century, the information flow of genetics and the structure of genetics were not the same thing. Now we know that genes reside on chromosomes and that your chromosomes move around and you have genes on there. And that didn't come to us. We didn't figure that out until like 20 years into the 20th century. At the time Mendel does, he didn't know anything about that. So all he had was information flow. That's all he had. And then the other thing that he knew was the plant thing. Remember we went over the little horticultural paintbrush thing? All he knew was that you had the gold dust, the pollen on his paintbrush. And that there was something in there that had the information for, in this case, the purpleness, if it was a purple male. Then he also knew that inside the stigma or the carpel was the female part of it. And he also knew that whatever was inside there was the information for white. That's all he knew. And that the information was what was segregating from the male, in this case pollen, to the female carpel. What that was exactly, no clue. How that worked, no clue. Remember, you had no clue about chromosomes at this point, no clue about meiosis, no clue about mitosis or cell cycle or any of that. That stuff didn't start happening and coming together until the 1890s, which is about when they rediscovered Mendel. So having a very clear view of what Mendel didn't have kind of makes this law a pretty significant thing. It's actually a pretty significant conclusion to come to, actually, considering how little you had to go on. So Mendel, in a lot of ways, was way ahead of his time, which is the reason why we forgot about it. But, which is exactly what happened. So he published his paper, and there's still some dihybrid stuff that we're going to take a look at. But he published his paper, and then it just kind of disappeared for a while. And we didn't really do much with it. And then about 40 years later or so, a group of geneticists, a new generation of geneticists, rediscovered his paper and his work and read it. Along the couple of those, there's several of them. William Bateson was one of them. William Punnett was another one. Punnett Square. And basically what they did was the response that they had was, there's two responses. First of all, what they wanted to do is they took a look at Mendel's results and they wanted to say, this is great. If I did what Mendel did, would I get the same results in my system, right? So some of them were working on corn, some were working on 4 o'clock plants, some were working on humans, right? But they all had the same question. If I do what Mendel did, would I get the same numbers that Mendel got? In some cases, the answer was yes. And so they basically confirmed Mendel. And in some cases, the answer was no. Now, for the people who didn't have the same results as Mendel, they had basically two different responses to this. Some of them would say, wow, the results I got were nothing like Mendel. So therefore, this chump was wrong. So screw him, he's out. Mendel didn't know what he's talking about. I do. And then the other group would say, you know what, I didn't get what Mendel got, but I do know that there's enough people out there who do get what Mendel got, but I didn't get it. So my question is, what's going on in my system? How is my system different? Do I have a different set of things going on? And I want to figure out what I'm doing. The people in the former class are forgotten to history. And that's almost always the case, which I always like to say is a scientific cautionary tale. Whenever somebody, when you disagree with somebody, don't call them a chump, right? Don't basically blast them, because usually when you do that, you say, oh, well, this guy's a fool. He has no clue what he's talking about. His data is worthless or whatever. When you're like that, when you're critical like that, like just because that's who you are, because you're an a-hole, Usually that's coming from a place of arrogance, right? That's not coming from a place of scientific honesty. That's coming from a place of arrogance and insecurity. Those people are almost always lost in history. The people who then take the latter approach, you know what? I didn't get what Mendel got, but I want to dig a little bit deeper into what Mendel actually got and then foil it against mine. And I want to dig more deeper into mine and see if I could use Mendel's techniques on mine and then try to figure out what's happening in mine. Those people are the ones we read about in the textbooks now. So I always like to stop and pause. That's a cautionary tale, right? It almost is never productive or advised to basically just snark at somebody because you're critical, right?
Examples of who, like,
Punnett, Bateson, Morgan, Sturdivant, Bridges. These guys were all in charge of Gerard, who worked on humans. I mean, all of the people who basically, we'll see Gerard later on in the next chapter. But I mean, the people who are in our textbooks as sort of forefathers who sort of took Mendel and then ran with it. Those are all the people that we grow up reading about, learning about. The people who said, oh, Mendel's not right, so obviously he doesn't know what he's doing. This obscure Austrian couldn't possibly know what he's doing. Those people are almost always forgotten in history.
He's a monk.
He was actually a monk. Yeah, like a Catholic monk. He was actually a monk. Yeah, only he didn't quite have as much issues with the Catholic Church, but this was well after the Reformation. But basically, when you look at the Punnett square, what you see is an organizational tool that actually Punnett came up with to sort of organize the crosses. And so if you take a look at a Punnett square, what you'll see is if we kind of do the double heterozygote cross like this, in the Punnett square, basically what's going to happen is on the top, you're going to put the gametes of the male, right? So the gametes of the male basically can only give either a big P or a little p. That's what you have here. Your female will either give a big P or in this case, a little p. So these are the gametes here. So in a Punnett square, basically what you do is you do all the possible matings. You don't know which one you're going to get, but you want to basically flesh them all out so you know exactly what you're going to get. No surprises. Here's what you're looking for. So if you take this pollen and this egg and you mate them together, what you're going to get is big P, big P. Take this pollen, this egg, you're going to get big P, little p, that's a heterozygote. Take this pollen, this egg, you're going to get a big P, little p, that's a heterozygote. Take this one and this one, you're going to get your homozygous recessive. Okay. So these are all your possibilities. And so you kind of can sort of diagram out what you can expect from any given cross. Now, if you are like an organism like corn, for instance, where you have lots of different offsprings, or each one of those kernels is a little offspring, then you're going to see all of these outcomes in their relative proportions. If you're, however, humans, right, where we only have one at a time, then what you're going to see is one of these possible four, right? So that's the reason why we tend to work, when we're looking at genetics, we tend to work with organisms that have lots of offspring, is so we can see all the different outcomes. When we're working with humans, it kind of takes a slightly different approach, right? With humans, it's basically more like, well, you have four potential chances, but your kid is only going to be one of those four, right? So the chances of you getting one of those four is 25%. So we look at things a little bit differently that way with humans. So here's kind of like the cross with the Punnett square. So here you can see you got your true breeder for white, your true breeder for purple. When you cross them together, you get basically a field of heterozygotes that you have one. When you cross your heterozygotes together, you can see you got your big P, little p gamete, big P, little p gamete. And then of course you have your 1 to 2 to 1 ratio, right? So you have your homozygous dominant, your homozygous recessive, and you have your two heterozygous. That's your 1 to 2 to 1 genotypic ratio. But you also have 3 purple to 1 white. Now, the nice thing about this, and the reason why this is powerful, is because like when I was at UC Davis in a fly lab, one of the things we do like we do what's called a mating scheme. So we basically set up like a big mating scheme, and we essentially sit down with pen and paper and we just write it all out. So if I took a fly of this genotype and crossed it to a fly of this genotype, then these are all the outcomes I should expect, right? And then I'm like this one, I'm looking for that exceptional version, that virgin, and I'm going to grab her and I'm going to set her aside because I'm going to mate her with a specific genotype and I'm going to get another batch of outcomes, right? With all the relative proportions. And so before I do anything, eventually then I'll get my data and then hopefully I'll be able to collect information. But basically before I even grab the first vial of flies, I have it all mapped out on paper, right? And this is basically going through all these genotypes, these alleles, through my Punnett squares. But here's the important thing, and this is oftentimes something that's missed in general biology, which shouldn't be missed. What you write out on paper is your theoretical, what you think is going to happen according to theory. But then what you actually cross in the vials is actually reality, is actual. And not all the time does actual mirror theoretical. However, when you set up a cross like this and you write it out on paper, not only does it get close to actual, but it pretty much for the most part predicts your actual. So I basically can write out my mating scheme before I even set up the first flyer, before I even plant the first seed, and I know exactly what I'm going to get. I plant the seeds, I do the crosses, and what do I get? The exact same thing that I wrote out on paper. So my theoretical is actually predictive of my actual. That's what I expect. And that's a very powerful thing, because that basically means that your theoretical, if it's that strong and can predict what actually happens before it even happens, that is basically proof that your theoretical, the way you think it goes, is you're spot on. You are accurate and you are correct. So your actual and your theoretical match up. It means your theoretical is accurate. So now let's take a look at the next cross that Mendel did, which is the dihybrid cross. In this case, what he's doing is he's using two separate traits in a single cross. And in this case, he uses, I believe it is yellow, green, and smooth wrinkled. So these are the two true breeders. And of course, he crosses them to create your double heterozygote in your F1. And then he crosses the F1s together to create, in this case, in the F2, a 9-3-3-1 phenotypic ratio, where you have 9 round yellows, 3 round greens, 3 wrinkled yellows, and 1 wrinkled green. Okay, that's great, but that made no sense at all. Right. So let's add a slide then. Because we're going to roll out the dihybrid in hopefully a little bit more of a clear way. I'm going to build it and construct it from scratch so that you can kind of see it as it happens. So you're still going to use your parental class, right? In this case, what I'm going to do is I'm going to use A and B, just fictitious genes, right? So capital A is going to be the dominant A, whatever that is, right? So the dominant phenotype. Little a is the recessive phenotype for A. B is going to be the dominant phenotype, obviously for B, and then little b is going to be the recessive phenotype for whatever little b looks like. So what I'm going to do is I'm going to create the true breeders. I'm going to cross the true breeders in the parental. So I'm going to go big A, big A, big B, big B. So that's a true breeder for both of these, right? Now it's important to understand that there's also alternatives to this. I can also still have true breeders if I do little a, big B, or little b, excuse me, big A, little b. Why? Because you're a true breeder for the gene, and these are both true breeders for genes, right? In the second case, I'm a true breeder for little a, but I'm a true breeder for big B. In the third one, I'm a true breeder for big A, but a true breeder for a little b. These are all 3 true breeders, okay? But for sake of simplicity, I'm going to stick with the first one. So I'm going to cross my big A with little a, little b. Those are my true breeder parents. So then what I'm going to do is I'm going to cross these guys together to create my F1. And that's going to give me an A from one and a little a from the other, a big B from one and a little b from the other. And then I'm going to cross this to another F1. This is the compound heterozygous of both genes. The important thing to understand in a hybrid cross is that the assumption here is that both genes A and B are independent events. That is to say that what happens at A has no hold on what happens at B. It's kind of like if I flip a coin with my left hand, does it have an effect on the results of my flipping a coin in my right hand? They're independent events, right? That's the same thing here. Now, in order to create your F2, you have to basically get gametes from these two parents, right? So how do you know what the gametes are? Well, first of all, each gamete has to have a representative from each gene. So that means each gamete has to have a selection from A and has to have a selection from B. So how do you do this randomly? Well, let's take a look at this. If I choose to select, first of all, big A in my gamete, I can have two potential pairings from the B gene with this. I can either pair it with a big B, that's going to be one gamete, or I can pair it with a little b. That's going to be another gamete. Alternatively, what if I choose little a in my gamete first? Well, then I can pair the little a with a big B, or I can pair the little a with the little b. The same thing is also true for the other parent because they exactly the same genotype. So they'll produce the same gametes. Now I'm going to draw my Punnett square. And now we're going to pair them up. Notice now you've got 16 squares. You went from 4 to 16. If you do a trihybrid, A, B, and C, then it turns into 64. So you can see that Punnett squares get a little unwieldy very quickly. Right? So you can't analyze your entire genome with a Punnett square. That would be like some astronomically huge Punnett square. So let's just go ahead and take a look and see what we got here, right? So let's go ahead and start pairing these guys up. So if I pair the first two together, I'm going to get big A, big A, big B, big B. Then we get big A, big A, big B, little b, big A, little a, big B, little big B, big A, little a, big B, little b, big A, big A, big B, little b, big A, little B, little b, big A, little a, big B, little b, big A, little a, little b, little b, big A, little a, big B, big B, big A, little a, big B, little b, little a, big B, big B, little a, big B, little b, big A, little a, big B, little b, big A, little a, little b, little b, little a, big B, little b, little a, little b, little b. Here we have it. That was easy. Okay, now let's do some counting, shall we? Let's take a look at phenotypes and let's list them over here. The first phenotype. What is the phenotype of that individual right there? Double dominant, right? Dominant for A and dominant B. How about the next one? Same thing, right? Still class 1. Second one, third one. Still the same, right? Fourth one. Yep. How about the next one down on the next row? Yep. How about this one? So it's going to be an A, it's going to be what? So it's going to be, so yeah, right? So it's going to be dominant in A, right? And then recessive, so we got one of those. How about the next one? So it's class 1, right? Last one in that row. So that's gonna be class 2. Now third row first. So that's gonna be a class 1, right? Next one. Take a close look. Yep, so it'd be 1, right? Okay, next one, third one. So that would be a different one, right? So now it's recessive dominant. Okay, got that one. Next one. So it's the same one, class 3, right? Okay, bottom row. So that's going to be class 1, right? Next one. That's be class 2. Next one. And the last one. So it's going to be double recessive. So what's my 9, 3, 3, 1. This is diagnostic for a dihybrid cross. By the way, this only happens if both genes randomly segregating. This would happen if the events of A had any bearing on the events of B. You would get a different ratio. So what Mendel saw basically was that not only do you have segregation, but you have random segregation, that these genes are segregating randomly from each other, because otherwise you wouldn't get a 9:3:1 ratio. That's only going to happen if it's random. So what's the importance of this? Well, first of all, setting down segregation was a big deal because we didn't. The second thing is the dihybrid cross. Now all of a sudden, for the first time, he's laying down that there's a lot of randomness associated with this process. Now we just came off a chapter where we're talking about a lot of randomness, right? But it's like hardwired in there. It's like intentional randomness. It's like you're wiring that in there for the sake of generating variation. Well, that's kind of where this goes, right? So he's basically, this is the first time when we actually get this hint that there's a lot of randomness. These things are doing their own thing randomly. So it's the first time we enter into the possibility of statistics entering into genetics, which is really kind of cool because when you read Mendel's paper, you can actually read all the elements, the beginnings, just the kernels of just about every major field of genetics. Like population genetics, you can see the quadratic equation actually in his paper. You can hear statistics in his paper, and that kind of is so. So he kind of discussed a little bit, initially set out some of these initial principles, and then in the 20th century, we took and ran with it. Question. Segregation, he gets all the credit for segregation and independent assortment. And for the work that he did with the ratios and things like that. So yeah, that was all him. Nobody else figured that out, or was even close. So this was a situation where he really did kind of figure it all out pretty much on his own. So like even with evolution, Darwin didn't figure that one out on his own. Wallace, whom many people forget, actually was at about the same time coming up with the same ideas. It's just that Darwin's the one that got all the notoriety. But for the most part, Mendel is the only one doing what Mendel was doing, because everybody else, like Darwin, for instance, was failing at genetics, right? Because he was the one doing snapdragons. Darwin was initially, and he failed at it miserably. The flowers, that's the one with the pink, the red and white, it's pink. That's that one. Yeah, he did that one, and that's why he failed, because he didn't actually have the scientific rigor necessary to sort of tease a complex problem like this apart. There's many things that Mendel did right. some of which were just luck, some of which were intentional, right? Another important thing that of science and Mendel's, there's a lot that you can learn about how to do science in Mendel's example, but one of them is reduction, right? Is you take a big problem and you don't attack the big problem, you take a little itty bitty piece and you attack that one and you try to make that possible. You try to reduce reductionism, the number. But now probably can. Uh, Okay. So, well, Zoom is apparently gone. So Elena and Heaven, I apologize. The last 30 minutes or so, you will not be able to see, but you'll be able to hear, nor will it be recorded. So, this piece here is probably not going to be recorded, so we'll move forward as we must. Okay. So this particular image now we can see with the chromosomes. This is basically kind of what we know now. And so you can see, in this case, they kind of overlay it. And I don't really like these next couple of slides, honestly, because what they do is they basically take what Mendel did in his dihybrid cross, and they basically superimpose over it the chromosomes. So what they do is they basically fast forward to like the last half of the 20th century, and then they overlay it on Mendel, which is fine. But the problem with that is we haven't been talking about that. So now all of a sudden they just dump all these chromosomes all over the place. And a couple of things, right? First of all, we didn't have the chromosome linkage to genes until like 2 decades into the 20th century. We didn't actually pin down really that DNA was the actual molecule of inheritance for like almost half of a century. So to basically superimpose this on top of Mendel's work is a little artificial. This is kind of like taking, but this kind of shows what it is now, right? So now we know that these alleles are basically going to be found. Oops, can't move it around. Sorry. Different system. Yeah, I know, right? Basically, they're found on chromosomes. So here you have your gametes, right? And then, of course, your heterozygotes, you got one of each, right? One from mom, one from dad. And then, of course, these are the gametes that we have from our cross. And then this is our F2. So you can see basically that our F2, which this is kind of what Mendel saw here inside the Punnett square. Then the F2, you can see these are the gametes up here. So you have each representative of each of the gametes themselves. So the idea there is that basically we will actually move our alleles around on our chromosomes, and they have a place to live on the chromosomes, specific areas that we refer to as loci, right? So a locus is basically where a gene lives. And ever being, I'm not going to give up on this.
Recording in progress.
Hear this mic. Okay. So this one's probably going to be a mess, actually, when you guys look at it. Okay. That's like really strong feedback. So these mics must be. Okay, better? Yes, I love it. Resolves, right? Resolution. See, tension, resolution. We talked about that during the semester, didn't we? Right. Okay, so now since we're talking about randomness, we got to talk about probability, right? It gets to statistics. Yay, right? We all love statistics. There's really only 2 rules in genetics. Well, there's actually a few more, but you wouldn't get to them unless you took the genetics class. There's not a lot of statistics in genetics, at least in the intro genetics class that we have in the fall. You got to get into upper level genetics to really start getting into those statistical weeds a little bit. But we have 2 major principles of genetics that we have that we took a look at, especially at this level. The first one is called the rule of addition. And in the rule of addition, what's happening is you're taking 2 mutually exclusive events. You're asking for an outcome to happen in either one, right? So in this case, what happens is you have 2 different events, A and B, and you want an outcome to happen in either A or B. You don't care which one, right? You want it to happen in either one, or A or B. So it's kind of like saying, okay, I have 2 quarters and I have 2 flips, right? I want a heads to happen. Doesn't matter if it's in the first or the second, right? I want it to happen in either one. So in that case, you use or the rule of addition. Now remember, sorry, that was not me, that was me. So basically, if you remember, or is the word trigger for addition, right? Remember when you're kind of decoding word problems, whenever you see the word or, it means that you use the mathematical operation of addition in order to get what you want. Now, how does this work? Okay, let's go ahead and take a look at this. Here, we're going to use our heterozygous cross, right? And here's the question. What we want to know is the probability of producing a heterozygote. Well, here's the or part, right? We have 2 different possibilities here, right? Because we have 2 heterozygotes. So one of them is a situation where dad will give the big P. So what is the probability of dad giving a big P? Well, for dad to pass forward a big P. It's 1/2. He's only got 2, right? He's got a big P and a little p. So it's a coin toss, right? So in this case, his probability is 1/2. Now, what's the probability of mom giving a little p? That's 1/2, right? So then the probability of a big P, little p is 1/4. Okay, we'll talk about that later. So that's the individual probability of getting the little p from dad, or from mom rather. But what's the opposite then? So what if we got the big P from mom and the little p from dad? What's the probability there? It's the same, right? It's 1/4. So what are you asking for here? What you're asking for really is, what is the probability of getting a little p from mom or dad? That's the or, right? So now this is assuming that the other one is going to be a big P, right? Not getting a little p from both. We're just or either one. So it's going to be the probability of each added together, right? So you have 1/4 plus 1/4, which means you're going to have a 1/2 chance. Yeah. So if you take a look, you can also see this on the Punnett square. Why? Because if you set this cross up, you're going to get your homozygous dominant, yes. Your heterozygous, homozygous recessive. How many heterozygotes do you have? 2 out of how many options? And what is 2/4? 1/2. So you can see the Punnett square actually sketches out your probability. So that kind of leads us to Mendel's second big law of genetics, and that's independent assortment. So basically what this essentially means is that not only are your alleles segregating, but they are doing so independently, right? That there are basically independent pairs, and they're doing so independently and randomly. So that's a big deal. That's what gives you that 9331 cross. So what's our next rule? A rule of multiplication. So this one is slightly different, right? You still have 2 different events, 2 independent events, A and B. But now what you want is an outcome, a specific outcome in both. So let's say, for instance, I want an outcome in A and B. Let's take a look at the quarter. In this case, you're flipping 2 quarters still, right? But now you want to flip heads in A and you want to flip heads in B. So what's the probability of flipping heads in the first and the second? So the and, if you recall, is the word trigger for which operation? Multiplication, okay? So basically, if you say, what is the product of 2 and 3? Well, that's a multiplication, right? Well, not only is product a multiplication, but 2 and 3 means 2 times 3, right? If you recall that from your word problem dissection work. Way back. Yeah. Nice. So I see that you guys sat together so you could like, it's kind of like you guys the kid look. Nice. So basically, when you take a look at your crosses, then we're going to use the same heterozygote cross, right? And so now you're basically saying, what's the probability of getting a little p? So this you would do the probability of getting a homozygous recessive, right? So the same thing is there. What's the probability of getting a little p from dad? Well, you've only got 2 from dad, right? So it's going to be 1/2. The probability of getting a little p from mom, well, she's only got 2, so it's going to be 1/2. So in this case, to get a little p from dad and a little p from mom is going to be 1/2 times 1/2, which is going to be 1/4. Now let's go ahead and take a look at our Punnett square. So how many homozygous recessives do we have? 1 out of how many options? So that is our probability. So another thing that we commonly do in genetics, what's called a test cross, right? Because it's not uncommon when you're working with phenotypes, for instance, and you're doing crosses, that you'll run across something and you'll be like, okay, so here I've got the dominant look to it. But guess what? I don't know what that second allele is. Because it could be 2 things, couldn't it? That second allele, which is an unknown at the moment, could either be dominant, homozygous dominant, or this could be heterozygous. So how do we determine the difference between the two? Well, what we do is we cross it to the homozygous recessive of that trait. So we basically set up our Punnett square. And we set up the recessive. So if this is dominant, then what's going to happen? It's going to give us different outcomes than if it's heterozygous. So for instance, in the dominant trait, it's going to give us all heterozygotes. So we're going to have 100% dominant trait. If it's heterozygous, it's going to give us a 1-to-1 ratio of dominant to recessive. We do this a lot, actually, because as we're sort of doing crosses, we get out some sort of a dominant phenotype and we're like, which one are you, right? I don't know what you are. You could either be homozygous dominant or you could be heterozygous. So which one are you? So we do to figure that out because we don't want to use that for our results, right? Because if we don't get the right one, that could screw all of our crosses up, right? It could be a pollution. If we're trying to look for the true breeder, then that heterozygote is going to pollute all of our work and it's going to screw everything up. So what you do is just basically take a homozygous recessive and you cross it and you can flesh it out. Now, why do you use homozygous recessive? And that's a question oftentimes will come up. It's very simple because you know it's there. Because you know homozygous recessive only has the recessive allele. That's all it has. So any difference that you see in your outcomes is going to be attributed to this allele. Why? Because you know what that is. You know what that is. You know what that is. Any difference that comes out is going to be attributed to that unknown allele. Not only that, but if you use a dominant phenotype, you have the same problem. You don't know if it's homozygous recessive or if it's heterozygous. So it's going to further compound your problem. It's going to make it even more confusing. Kind of what it looks like. So here you can see this is basically what you expect if you have a homozygous dominant, all the dominant trait coming out, versus if it's heterozygous, you're going to see a 1-to-1 ratio. So even in genetics labs, actually today, we still do test crosses so that we know exactly whether we have a heterozygote or if we have a dominant homozygote. We can also do this with dihybrids, right? So for instance, this is going to breed true for both A and B. This one will only be true for B because it's heterozygous in A. This will only breed true for A because it's heterozygous in B. This one will not breed true for either because it's heterozygous in both, right? So just a little bit of 20th century component here. Basically, when we take a look at DNA, we now know that thanks to the 20th century, this is later half of the 20th century, that DNA is essentially our informational molecule for the organism. We also know The DNA is sequence that encodes directly amino acid sequences for proteins that we need that will then add to our phenotype, right? So like hair color, for instance, our pigmentation is a protein that's built by our pigment genes. That also basically leads us then that if we can alter the genetic code, we can also alter the amino acid sequence, which means we can alter the phenotype. We do that by mutation. Right? Mutation is basically an alteration in genetic sequence. So you change the code, you change the amino acid sequence, you change the protein, you change the phenotype. Here's the problem. Generally speaking, mutations are mostly deleterious. That means they'll cause death. Like by far, like not even close. They are mostly deleterious. And not only that, but mutations, when they happen, are acted on by a natural selection. And of course, then that will basically lead to evolution. But here's the problem. This drives me back because you get this from the time you're little. It's the sock puppet version of evolution. Oh, we have a mutation and then we turn fluorescent pink and we're all pretty and that gives us an advantage because we're pretty or we're blinding to the predators, right? And so natural selection selects us. And so then we all become pink and we've evolved, right? Aren't we all happy now? That's bunk, right? Because the majority of mutations are deleterious. So oftentimes what you're getting in the sock puppet version of evolution is kind of a skewed view of things. You're looking at beneficial mutations because we know that we need beneficial mutations in order to create this. The problem is the overwhelming majority of mutations we get are actually deleterious. I mean, by a lot. And here's the thing. As a matter of fact, in the genetics class, I always kind of show them a book that I've got that was published in '88. One of the last times they published it in print. It's a catalog that's like about that big. And it's in my office actually still. I always show it to them because it's gotten so big now that it's now online. It's actually online in a Mendelian Inheritance in Man. It's a catalog of all of the genetic disorders, human genetic disorders that we know of at that point. And it is like an overwhelming list of like human suffering, genetic catastrophe, and absolute devastation. That's how big the book is. And that's just what we know. So I always use that as like an example of like, okay, when we talk about mutations, this is mostly what you get. Absolute train wreck disaster, right? So the frequency, even the most, and there's different mutation rates that are published. And the reason why this is important is because mutation itself is a major mechanism of evolution. But the problem is it has some titanic caveats to it because most mutation rates that are published are grindingly slow. And that's just mutation rates in general, right? We're talking about one in every maybe a couple 100,000 years, maybe to 1 million, depending on the mutation rate that you're looking at. But then that's just mutations in general. The majority of those are devastating. So you would see negative selective pressure. So you would basically be eaten first and your genes would never make it, right, to the next level. So you would be omitted from the gene pool. That's not evolution. That's omission, right? So what we're looking at is beneficial mutations. So the beneficial mutation is even further apart. So the mutation rates are even slower. So there's some like, and as a matter of fact, if you kind of calculate it out, just sort of roughly and there's a lot of ballparkishness in there because there's a lot of variance in these mutation rates. But if you just kind of take a look at it, you're thinking, okay, wait a minute. So first cell came around 4.5 billion years ago or so. In order to accumulate enough beneficial mutations to create this much coordination and regulation, is going to require a lot more than 4 billion years. It's going to require something on the order of 14 to 20 billion years, which is actually getting to the age of the universe, right? So you're talking about a really significant problem here. And that's the reason why I'm not a Darwinian gradualist, right? But at all, because the gradualist idea, I think, is actually catastrophically wrong. And it came from geology, and the geologists are wrong, right? Because they're a little weird. I mean, if rocks are your best friends, I'm sorry, pet rock notwithstanding, it's not right. But the reason why I say that is because this is important, right? Because you have two different camps in evolution. A lot of people don't realize this, right? You have the gradualists, who are the ones that said, basically, geology-wise, that all of the processes you see on Earth, those slow glacial depositional rates that we see, the slow rates of erosion that are very, very, very slow, that these rates were in play for infinite past and infinite future. And then basically what we see now has been sculpted by those rates. They haven't changed ever in the past or ever in the future. That's a theory called uniformitarianism. It comes from geology. And the problem with that is it's absolutely wrong. It's actually flamingly wrong, because we know that's wrong. And I pick on the geologists because they're the ones who were the most nearsighted about this, right? Because they basically created this gradualism based on deposition rates and things like that. And then this is how we can sort of, like a clock almost, right? So if we have deposition rates, then we can sort of calculate backwards how long it took for this mountain to erode, right? And usually you're talking about hundreds of millions of years and things like that. And so we could use this sort of as a clock. And they were unyielding early on in the 19th century, by the way. They were unyielding and basically saying that there's no room for catastrophe, right? That the idea of a catastrophic event having short bursts of massive environmental upheaval and rate and change, is there was no room on the table for that for geologists. Now, the problem is we know that's stupid, because we live in an era where we have gone through St. Helens, where we saw an entire mountain blow its north face off in a matter of seconds. We've seen in the canyons of St. Helens, entire canyons being carved out, not by the slow creeping erosion of a river, but by within weeks being carved out by these violent mud flows as they flow through these canyons. So we've seen catastrophe happen. You know, we've talked about Chicxulub, right, which is the big meteor that hit the Earth 65 million years ago and killed all the dinosaurs. When that first came out, that was super controversial. Now it's kind of like a yeah, duh moment. But back in the day, I remember when I was in school, that was like, no, that's not possible, right? Because the Darwinian gradualism said the dinosaurs died off slowly, incrementally over hundreds of millions of years of time, were slowly replaced by speciation of mammals and things like that. Chicxulub said, no, your gradualist timeline is screwed up. Catastrophe happens, and when you have extinction level events, then it causes massive die-offs, and in populations that survive, go through massive bottlenecks, and then that changes everything genetically. And so we just now kind of come to grips with that. And so not only that, but in the fossil record, it had issues of its own, right? Because the idea was, if everything is moving along incrementally, the way the gradualist said, then where's the missing links? You should see lots of transitional forms. That's not just a pushback. That was actually true. There was a problem there. The fact that you saw fully formed organisms and then fully formed organisms here and nothing in between, it's like, where's your progression, right? You should see your progression here if everything went incrementally by steps, gradualistically. That was such a problem that in the 70s, Stephen Jay Gould and Stephen Elledge came up with punctuated equilibrium, which is an alternative to gradualism. And basically what this said is that for the most part, the world is in states of equilibrium where gradualism seems to be at play. But it's punctuated by episodes of massive catastrophe and upheaval, where you have incredible environmental change in a very short period of time, like St. Helens, for instance, on a larger scale. But it's accompanied with massive mutation rates that increase dramatically, and speciation rates increase dramatically. So it increases so dramatically that you don't ever see the transition there because it goes so quickly to the apex that you see the fully formed organism, and then you hit another plateau of equilibrium. And so that basically puts a place in there for catastrophe, which we know happens all the time. Matter of fact, Chicxulub, which we all know and love, is actually only the fourth largest impact crater on the Earth. There's been bigger, actually. Yeah. So there's a lot, right? So there's a lot of catastrophe on this planet. So geologists, basically, I think when you look back at it, we're kind of like, boy, you guys are stupid for just putting this off for so long, right? Because I mean, it's a thing. That's kind of what I like to mention, because mutations, right? It's got issues, right? And you're not doing any favors by saying, oh, well, it's perfect. You get all these nice beneficial mutations. It's like, well, it doesn't really work that way because we don't have enough time on this planet to have accumulated this many beneficial mutations. This didn't work that way, right? So if we've only been here for 3 billion years, what that means is the mutation rates had to have been different if you're going to be accumulating beneficial mutations. There's no other way around that. And that's the where punctuated equilibrium comes into play, right? As it turns in now, the world we're living in now is essentially a combination of punctuated equilibrium and gradualism. But now we want to do is we want to talk about extensions to Mendel. So what I want to do is I want to basically highlight what Mendel did first. Use the next couple of minutes to do that. We're going to take a look at what's called patterns of inheritance. These are basically rules by which genes segregate. Now, we've already covered one, the Mendelian pattern. So the Mendelian pattern has 4 major pieces to it, right? So what Mendel saw in his pattern was that, first of all, you had one gene for one trait. There weren't multiple flower color genes. There was one gene, right? And in this one gene, you had 2 alleles. You didn't have 2 genes. You had 2 alleles in a single gene. He also noticed that you have complete dominance. Right, so you basically don't see the white when you have purple present. And the other one he also saw was random segregation. If you basically follow all those 4 criteria, then you will segregate according to Mendel's rules. You will generate things like a 3-to-1 ratio. You will generate a 1-to-2-to-1 genotypic ratio. You'll generate a 9-3-3-1 dihybrid ratio if you follow these 4. However, we have violations. Let's violate this, shall we? The first violation is going to be with polygenic inheritance. What is polygenic inheritance? Polygenic is basically where you have one trait driven by many genes. Genes. It's something that we oftentimes will refer to as continuous or quantitative traits. If you get to quantitative traits plus environment, like nature and nurture, we call them multifactorial. But the reason why they call this continuous is because if you have a lot of genes, what it does is that your phenotypes will create a bell curve. You'll have rare outliers on the tails. You'll have a lot of individuals in the middle and another extreme, but rare extreme on the other side. Okay. This is what's called a continuous distribution. It's the bell curve, right? And the more genes you have, or the more variables you have, the more of a bell curve it creates. Okay. Now we're going to pick up with this slide here Actually, let's just do plyotropism. I just want to show you an example, right? Because here's height, for instance, right? Height, you don't have categorical. You don't have 4 foot, 5 foot, 6 foot. You have everything in between. You have 4'2", 4'3", 4'4", everything in between. So if you line up by height, you can see you've got your bell curve right here. So this is basically driven by multiple genes in your height. Weight is another one. Skin color is another one. These are all wherever you have a big bell curve. Very few very light people, very few very dark people. Most people are somewhere in between. Okay. So we'll talk about pleiotropy next time.